Today it's a pleasure to have Dr. Tuğrul Giray here. He's a METU alumnus living in Puerto Rico. He has a BS from biology at METU. I'm not going to tell the year. Should I tell? 1990? Sure, okay. He has completed his MS and PhD in entomology at University of Illinois. He has also completed postdoc work at Smithsonian Tropical Research Institute in Panama in 1997 and at University of Vermont between 1998 and 2000. He began his current position as an assistant professor in the Department of Biology at the University of Puerto Rico in 2000. Currently, as a professor of University of Puerto Rico, he collaborates with laboratories in different countries to study honeybee populations, their health, genomics, and behavior. This research is funded by grants from international, federal, state, and local organizations. He has over 100 publications in peer-reviewed scientific journals, edited books, and popular science journals. He mentored 20 graduate trainees from around the world who are currently working in academia, industry, government, and NGOs in several countries. Without further ado, I'd like to leave the stage to him. Thank you.
Thank you very much. Thank you for the invitation. I'll just jump into the talk. So these are the institutions that I have been affiliated with. Today I will talk about individuals and the unit that they form, the hive. It's important to look at traits of individuals versus groups. One indication of that, an example, is with this chicken and egg story. It's not who's first, who's last. It is like, what should we do to increase, say, egg production by chicken? When we try to select the individual chicken chicks that produce most of chicken that produces the highest number of eggs, we are not very successful. It takes 8 generations to just increase from, say, 10 eggs in 10 days to 11 eggs. If you do a group selection, look at the group, see if the cage is producing more eggs than the next cage, and use those as your parents, you accomplish in 4 generations even a greater success. Go from 10 eggs in 10 days to 15 eggs. And on top of that, you also reduce the aggressiveness of those individuals. So the reason this works is because chicken are not packing each other, trying to hurt or for access to food or kill each other. So we can avoid cutting the beaks of chicken. As you see there, that individual has the beak cut, and you have to do that for all the chicken in your production if you have not selected for group behavior. So that sort of tells you the importance of this. This work was published in Science, Poultry Science. When we look at bees, which are a lot more integrated, it becomes really important to look at group traits. There are individual differences. Here you're looking at a hive where you have the queen in the center, workers around it, and then you have some individuals dealing with honey, others, a male over on the right hand up top. So they form, although they are individuals, form this cohesive society that works towards one end. We are trying to understand how those different individuals form a functional society. That's the main question that I am after. This is an excerpt. What I took here is from Dennis Wells. He's a filmmaker from Germany that was interested in our research. I talked to him a lot about bees during his first film, so he made a second film on honeybees. This is from that film.
Our lives are measured in wing beats. I'm a forager bee. We need to collect 1 kilogram of pollen and 2 kilograms of nectar every day. And my colony is depending on me. But the world is an unpredictable place. working, we'll make it through. Our colony is 30,000 strong and growing. We need a new nest, and we need it fast. One of us has to find the answer. It might as well be me.
So that film is based on life of a single bee as it goes through life, and it goes through reproduction, foraging, growth, all of that is included. And the focus is that single bee. Of course, it's not a single bee, you have the whole colony to look at. But it shows you what kind of challenges, the movie shows what kind of challenges a hive faces and how they are able to overcome this, from fighting predators to finding nest places. So here is the life story of that single bee. Starts as an egg, turns into larva, then adult, and adults first thing that they do is nursing these young. So that's the youngest group. Then there's the next group, age group, that does these different tasks. Some are guards, like a boxer standing at the entrance checking who's coming in. The next group is, this is glimmer of honey there. It's basically storer bee, took the nectar and depositing it in there. And here is an undertaker that's carrying a dead bee out of the hive. So that's about same age group. And the next phase is the most dangerous tasks, such as collecting nectar, collecting pollen, or stinging intruders, defending the hive. And there is plasticity between these tasks. So task groups are not static, so individuals can go back in what they're doing based on colony's need. Also, they can switch up and down between the different jobs of the same age, depending on the need. So we're trying to understand how this plasticity occurs and why in different bees this is different. So the long term is over weeks, and the short term change occurs within hours. To show you a little bit of that variation based on location, based on environment, I'll discuss Oskai's research. He was one of my first graduate students. Currently he's in Namık Kemal University as an associate professor, and he studied bees from the Syriaca bees from south, Mediterranean bees from Muğla region, and Caucasica bees from Artvin. These bees were put in a common garden, which is difficult to access currently, I understand, so here on the campus. And in June, you can see that Syriaca focuses on producing a lot of bees. There is no sealed brood, sealed honey. There's a lot of sealed brood. Caucasica, the northernmost bees, really have a lot of honey already accumulated in June. They are getting ready for winter that will come early. And Carnica has sort of more of a balance, the four-season bee. And we looked at those bees, two of them, Syriaca and Caucasica, in two different common gardens. One was in Ankara. So these are the Caucasica. Their color is also different because they need to gather heat, warm up. And Syriaca, the yellow bees. So we looked at them in Ankara, but also in Kırklareli after Devrim established his lab. So our hypothesis to understand how the hive phenotype forms was that all the bees form the hive phenotype. So if you have more honey, that means more bees are collecting honey. And then you will make less brood because that's not your focus. You're not trying to reproduce quickly. And you will have also more bees that are just because you're not swarming, you're not reproducing. You're accumulating. The bees that come up, they just work on collecting more honey. You have a bigger workforce. So this looks like indeed bees can explain the colony phenotype. We also looked at defenses, and turns out that in a tropical region, more subtropical region, warmer regions, you will have more of a longer season of brood, you have more diseases, so you have to have a better defense. And indeed, there were more bees that were controlling varroa mites by removing either the mites or cleaning quickly cells that were affected. So this again goes to the idea that yes, indeed, bee behavior may explain hive behavior. We wanted to see if foraging pattern was also different of those individual bees. To do this, we used von Frisch's method of color detection in bees, that they can tell color differences, and we provided them with 3 different colors, yellow, blue, and white, in these plexiglass, cut plexiglass flowers. So it's nothing comparable to artificial flowers that you guys are producing here in Metu, but that's our flowers. In this study, Meral Kence was our host. Harrington Wells was the person who designed this mechanism. Bees come here and they, of their own volition, choose one color or the other. The flowers are equivalent. They give the same food. Next thing what we do is we change the variation of food provided by the preferred flower. So if the bee likes yellow flower, we make it to not give food in 2 out of 3 visits. So we just put 1/3 of the flowers with food. Then when we look to see if they will change their decision, Syriaca bees do not change their decision. Either under constant reward condition or variable reward condition, they do not visit other flowers. They just go to the one choice they make. Whereas Caucasica, even under constant conditions, once in a while goes to about 10% of the time, checks what else is present. So those are the ones that have to accumulate a lot of honey. And indeed, if the food becomes sparse in one resource, they quickly switch to the other. So there are numerous bees, bee force that is collecting honey, plus they are going to collect from anything available. But we wanted to know what underlies this difference in plasticity. Okay, so we wanted to look inside the behavior of these individuals. So for that, Charles Abramson, our collaborator from Oklahoma, with Eddie Perez, an undergraduate student, then later he got his master's with us, and then currently he's doing on informatics and statistics PhD in Pittsburgh. So they designed this experiment on proboscis extension response conditioning. So what that is if you give an odor to the bee, like ringing the bell for a dog, Pavlovian learning, and then provide sugar water on a Q-tip or on a toothpick to the bee, quickly bee associates that odor, which is a conditioned stimulus. They need to be conditioned to that to respond. They start responding, giving the conditioned response to the odor only. Here, Okan did the studies, also Babur also did these studies, so you guys are familiar with Charles and the study. What we do is we give them 2 different odors. One we don't reward, one we do reward. So the bee will need to distinguish between the 2. And a self-respecting bee does learn during acquisition when you are providing food. And if you withhold food from that odor, then they stop responding. The attached line shows the response of, say, Ligustica or Anatoliaca. But for Syriaca and Caucasica, they did not quite stop responding. Even after 12 trials, they were still responding to the first odor that they learned. At the same time, the second odor is not rewarding, so they don't respond to it. And then when we do the switch and start giving the second odor, giving the reward, Caucasica learns, just like any self-respecting bee, but Syriaca insists on responding to the first, sort of resembling the color experiment. So now this is odor, not, it's an olfactory stimulus, but again we see this persistence and not changing in the Syriaca bees. We also wanted to see if we can sort of mimic more environmental conditions and balance effort and reward. So maybe if things are not as easy, there is more like natural flowers, we can maybe induce plasticity, because we are like, a bee has to be plastic. They should look at something else also. So for that study, Jenny Acevedo, who's currently at the Institute of Forensic Sciences Institute of Puerto Rico, worked with Jean-Pierre Aleman. At that point, Jenny was a graduate student, and Jean-Pierre Aleman was an undergrad student. We made these flowers with a modification. Of course, 3D printing, etc., could be better, but we used pins and cut them to make long or short pin flowers, long or short stamen flowers. So the design is you have 4 microliter, 1 molar sucrose in 36 flowers of alternating colors in the control phase. So bees go there and they should basically not care and go to either color. Blue and white are preferred pretty much equally by bees. In the acquisition phase, we reduce the reward of the white flowers. They are still easy to access, but we put then long stem and blue flowers with higher reward, 2 molar, 4 times greater reward. And then in the reversal phase, we reverse the color and reward association. So we want to see if the bees will go to, say, easy flowers and then again to easy flowers. So they will switch from white to blue or from blue to white, depending if they want higher reward. In Anatoliaca, this is how we divide the bees. If they are just going always to the same color, like the triangle shows, doesn't matter what phase, they are color constant bees. So they just preferred one thing and continued on that. If they are switching between higher molarity, they are going to basically blue flowers first and then white flowers next, then they are energy maximizers. If they are going to the easy flowers in both phases, then they are work minimizers. These are sort of equally divided in the Anatoliaca. In our Caucasica and Syriaca, it's not like that. Syriaca seems to be work minimizers. So they go to the easy flowers, and they do show some plasticity on this. Caucasica is more similar to Anatoliaca, but still there is an excess of energy maximizers. So they tend to go for higher reward, which is consistent with higher accumulation of resources. So although Syriaca and Caucasica bees did not show reversal learning in perconditioning, they showed reversal under more typical foraging conditions. So foraging strategies are equally represented in other subspecies, yet Syriaca bees are work minimizers and Caucasica bees are more energy maximizers. So what may be underlying this? We know that dopamine and octopamine, different biogenic amines, control preference and plasticity, especially dopamine. So we examined then brains of those bees. We dissected the bee's brain, separated different regions. The circle is around mushroom bodies, which is the learning and memory region, and checked expression of dopamine receptors in individuals before and after this foraging experience. And turns out that in that short time, in both bees, dopamine receptor 2 levels decrease in the brain. Probably more receptors are being produced, RNA is being consumed at that short time. However, the response is much greater in the Caucasica bees than in the Syriaca bees. So Caucasica seems to be sort of more reward and more plasticity with this biogenic amine. Octopamine is important for evaluating sweetness or evaluating sweetness of food. That was important for both of the groups, and there was no statistical differences between Syriaca and Caucasica for that. So we can focus on what adjustment has been changed to make a bee go after easy and constant food. Now, there are other organisms where such models have been developed. So I'll share with you one of the models. So sea slugs also show similar behavior, and dopamine seems to be the promoter of easy foraging or more difficult foraging or more plastic foraging. Okay, so brain gene expression of octopamine receptor and dopamine receptor in the mushroom bodies correlate with foraging experience, and octopamine pathway signal appears to be equally important for both subspecies. We also did octopamine application, and that just made them become sort of better work minimizers. Dopamine pathway signal expression change has a significantly greater amplitude for the Caucasica bees. We'll come back to that story. We cannot always work in Turkey, so because our lab is in Puerto Rico, so we looked at the Puerto Rico bees. My second graduate student, Bert Rivera Marchant, here shown in all attire looking at the hives, came up with this idea that, okay, bees are important, but you see the environment seems to be important. So how is the environment changing things? And can we predict something about what they do in terms of defense, foraging, pest resistance, the things that we looked at. So he is telling that in continental temperate regions, resource is restricted in time, has an abundance peak, but has a restriction. But in space, once it is available, it's all over the place. So you have blooming, spring, everything is in bloom. In continental tropics, though, you can move from one place to the other, like the gazelles going over Africa, and you're going to find food someplace at any one time. But over time, you can always find food, but it's not in the same place. You need to go to some other place. Whereas in our island, it's a hybrid where you have both time limitation and space limitation for where you're going to find the food. Because if the food isn't there, if you try to move, you will be in the sea, in the ocean, in a short matter of time. So that tells that you will have low defense when you basically need to worry about collecting food instead of defending the hive. Whereas it's important to defending the hive because you're going to always find food. Don't worry about that. Worry about reproduction. Defense will be high. And for foraging, again, we are estimating tropical islands will be more like the continental temperate regions. This is based on island biogeography. First, we wanted to see if we need to worry about different bees in Puerto Rico. So we went all around the island, sampled the bees. This is Alberto Galindo, the third student in the lab. He developed this method with a meteorological balloon and a queen tied to the balloon. You can attract the male bees at the mating areas. So that sort of combines a 1 square mile area, 1 square mile radius. You can then bring that balloon down and with a scoop, with a net, collect the bees and sample them for genetics. So with this method, we found that basically every single town is relative to has relatives in every other town. So it's really a single population of bees in Puerto Rico. And Jenny, the reason she ended up in the Forensic Sciences Institute is that she became very apt at doing genetics analysis of parenthood, and here she's demonstrating that the GAHB, gentle Africanized honeybee, is coming from Texas. It's right in the, from the 1994 Texas samples, Africanized bee samples, a single introduction, and then it spread and expanded on the island. So to answer this second addition to our hypothesis. Okay, the hive could be explained by the bees, but also by the environment. So you need to look at the sum of environments and sum of bees to determine what is happening in the hive. So I'm going to try to look if this idea that in the tropical island, indeed, there is a time limitation for food availability. People who come to the island say that, oh, this is all the time is summer. There is no other season. It's not true for the bees. Maybe it's not true. So Mehmet Ali Döke with Stefani Feliciano, they did this compilation of data on availability of flowers that are visited by honeybees. And indeed, there is a scarcity from October to January. Only about less than 20% of the flowers bloom in that season. And the rest of the season, 80% of the bloom is present. So there is basically the conditions necessary for basically having a winter-like period and an unavailability of food. Does that cause change in longevity of bees? Do we get something like a winter bee? And that also is true. If we look at March cohort versus October cohort in colonies, we find that the October cohort lives up to 3 months, whereas the March cohort lives less than 2 months. So we demonstrated that, but this is observational. This is not experimental, so we wanted to see if that has anything to do with reduction of brood, reduction of materials. So we did this experiment where in 5 different hives, we used one hive with uncapped brood, capped brood, like the regular hive demography. And then in our treatment colonies, we just did not provide them with uncapped brood. So they basically don't have the interaction with the young brood. When you do that, even though queen is laying eggs, she's laying it in a cage, so those cannot be attended. The cohorts of bees that are painted over time in these 2 colonies were observed. They are one-day-old bees. They are adults. They're not baby bees, so it's not a developmental, sort of long developmental, but more of a behavioral developmental response. And indeed, the treatment bees lived up to almost 4 months in these conditions, whereas our control bees, just like any self-respecting summer bee, died before 2 months. Here's a comparison of their physiology, so it's not just sitting around, but they're and not dying, but their physiology is different. Their hypopharyngeal glands, the glands that produce food for the larvae, remained large in these older bees in the middle of summer. So we found the mechanism, the signal from the brood tells how long you need to live and what you need to do. So to test the defense idea and availability of food, scarce food on space, we did 2 things. We looked at foraging to see if they were plastic in their foraging effort, and if they were only color constants or if they were varying their choice. We found that they are plastic. They are sort of like the Syriaca bees. This is work by Noel Fanfan from Haiti, who completed his master's with this work, continuing on to his PhD now, and showed that typically these bees are work minimizers. So he chose different colors to show the proportions of the groups, and it does have control again by dopamine. If we provide dopamine to the bees, treat them with dopamine, then we can shift them to be generalists, so they do not go to. But this is like an artificially heightened dopamine level in the bees. If we instead block the dopamine, reduce it, sort of make it a lot more like Syriaca, then they become color constants, mostly color constant bees. So they go to their first choice and then don't switch over.
How do you treat dopamine externally?
Dopamine is, we gave it, you can put it on thorax, that's the best spot, or you can also give it orally. that also works. So there is a study that's showing either way. Flupentisol application was only topically. And again, you can apply it on the thorax or you can apply it on the abdomen. Both work, thorax work better than abdomen application. So that's an antagonist of dopamine. So we block dopamine and see the opposite response. So how defensiveness-wise, we expect reduced defensiveness, but pest resistance basically depends on presence of the pest. So we compared in multiple studies the Puerto Rico bees with European bees, and I'm also plotting here other studies where Africanized and European bees were compared. So we have about 3-fold, 4-fold decrease in aggressiveness in these bees that are of Texas Africanized bee origin on the island. Their resistance to mites continues to be high, and they are biting there, a varroa mite. This we did it under light conditions, but under red light conditions, they still are able to find, for bees in complete darkness, they are able to find the enemy and bite it and cause damage to that. So our study summary was published in Evolutionary Applications. We then wanted to look at the genetics in the individual of this aggressive response. So we used the method developed by Maitreya Agarwal during her master's. She later completed PhD first went to Fukushima, but there was the leak, so had to go to India, completed PhD there and got a job there. So Maitreyi developed this shock assay. We used that for sting response. We increased the voltage. So that paper is this one. It may be of interest to some in the audience for the voltage response. And turns out that if a colony is more aggressive, bees the workers you take from there and induce them to produce sons, their sons are also aggressive. So there is a genetic basis for this behavior. How do we look at aggressiveness in males? They don't sting, they don't have a stinger, but look what happens when you give them electricity. They try to bend the abdomen and pretend to sting. So that response is what we looked at to correlate with the sting response of the female on the left. The person who noticed this behavior was Jocelyn Rodriguez Cruz, an undergraduate. Basically, Aryan gave a drone to this girl. Look, you can hold it, honeybee drone, it doesn't sting. And the bee did this behavior, and the girl, oh, she's stinging me. And that resulted in this nice paper. So okay, that's at individual level how we look at aggressiveness. How about the colony level? What do I mean by that? This is the example where you can see some real Africanized bees in Mexico. This is Dennis Wells. This is the first movie.
But in the end, the protection was sufficient to work among 40,000 angry bees.
So this is just one colony and just basically opening the cover. That movie is The Secrets of the Hive. Both of the movies are now on YouTube freely available. Those white specks are stingers on the leather patch. So you can take those and check genetics also of the bees that stung. And you can swoop bees that didn't sting and look at their genetics also at the same time. So this is an exaggeration for the movie, but really in 1 minute you get 1,200 stings. That's 150 stings are going to cause kidney collapse, and the person, the subject, will be dead if not taken to hospital immediately. So we use this method and look at colonies from around the island, collaborating with Gene Robinson. At that point, Aryan was a postdoc in Gene's lab. And Aixa Ramirez was my PhD student, who's now in Puerto Rico Department of Agriculture Aryan is at US Department of Agriculture. They are researchers. So we took pairs of hives from different municipalities, brought them to a common garden at our facility, and looked at their intensity of defense. We did this twice with sting assay and a behavioral observation. And there is a nice distribution, sort of like maybe normal distribution for hives. And we sampled in each colony 20 bees that were stinging and 20 bees that were not stinging. The connections between the bees show their relatedness. So indeed the colonies were separate from each other except for this colony 6 that is sharing some, maybe the same, drones from the same colony mated possibly with the queen of these two. We controlled for this relatedness effect and looked at alleles across the genome, across foragers and soldiers to see if we can find what makes a bee a soldier. And this is the result. The line shows you the log 10 p-value, that's the threshold for significance. So across the whole genome, looking at 2 million SNPs, 16 chromosomes, we got empty, nothing. Okay, so basically individual response does not explain colony level defensiveness. You need to really find the stinging bee or the bee that will sting and then get it sung, etc. So there are genes, yes, but related to aggressiveness, but not explaining the colony aggressiveness. So individual aggressiveness has nothing to do with that. Is one interpretation. So we looked at hive level. We looked at all the alleles in the hive, so it doesn't matter the frequency of the alleles in the hive in those 20 individuals, and see if that will correlate with aggressive response. And then we got many targets. Look at linkage group 7 is very much highlighted with a lot of single nucleotide polymorphisms that match aggressive response. Turns out that these has to do with biogenic amines or peptides that determine state in the brain. ECE2 was one of the genes that is a metapeptidase that produces the neuropeptides. And DPR4 is a defective proboscis response gene identified in Drosophila related to responding to chemicals. So because the defense depends on communication, pheromones released, colony response have to do more with are you a responsive unit more than if you are a defensive individual, aggressive individual. So turns out that worker interactions are important. So not only the environment, but now we need to add the interactions between workers to our model of the hive. So I think I'm going to stop right there. There are more studies to show, but we're out of time. We looked at the microbiota, and microbiota is important for development, antibiotics delay development. And to look at development in the lab, we used a circadian activity development. It develops over several days, and we can alter that. The circadian rhythm is controlled by these few cells that extend all over the brain, called PDF cells, and their numbers increase as bees become rhythmic, as the rhythm develops. That is a collaboration with Ricardo Scheiner and Katharina Baer from University of Würzburg. And what we did is we repeated the antibiotic treatment just to look at circadian, see if it is similar to foraging. And indeed, circadian rhythm development is reduced. And in the brain, PDF cells that we can see have reduced. So we also connected that all the way to the brain. It wasn't microbes that you receive from other workers, so the nurses had no effect on this. It is really the bacteria that you get from the hive, from the comb, when you are chewing the comb and coming out. Sort of like similar to the story of C-section babies versus babies that are born through the birth canal. So they are getting their mother's bacteria and protected against different things, and their immune system develops better, so no asthma, no allergies. So here it seems like this is really important for these bees. So our N definition then includes the biotic environment too. So to understand the hive, we need to know how the bees, how their interactions, how their biotic environment and their abiotic environment come together. Studying social and individual phenotypes of honeybees is what I call biology or biology. Okay, thank you.
Thanks a lot.
Thank you. So this slide is for people, I mean, it's incomplete, definitely incomplete, but the yellow bars show PhDs from the lab. If they have green also, that means they also did postdoc with us, or only postdoc. If it is blue, light blue, that means they did masters and then PhD in the lab. Thank you.